Hello and welcome to Utkalp Digital Classes. I am Richa and I am your mentor for the CSIR NET Life Sciences course. This course is specifically designed for you all to qualify the CSIR NET Life Sciences exam with a good rank. So the key to qualifying this exam is preparation, solving your previous year question papers or model test papers, and timing your preparation. So if you time your preparation, say you keep 1 hour and solve 70 questions of part B in this 1 hour, or you solve 25 questions of part C in this 1 hour, it is going to help you a lot in your exam. You'll be able to solve questions quickly and you'll be aware how much time you have to devote to each unit. So this lecture is specifically designed to solve some previous year questions. The lecture is going to include questions from this entire playlist. We are going to solve questions from Cell Biology Unit 2 of the CSIR NET Life Sciences syllabus. So, starting with the questions. The first question is, cell organelle covered by a single membrane is? So, which one out of lysosome, peroxisome, glyoxysome is covered by a single membrane, or is it that all of them are covered by a single membrane? So you have 4 options. I hope you are able to solve this question. The answer to this question is all of these. Yes, lysosomes, peroxisomes, glyoxysomes, all 3 are covered by a single membrane. They are not double membraned organelles. So here we are just going to tick all of these. So the next question is, which one of the following is not present in a plant cell? So there are 4 options of organelles given here and you have to choose which is not present in a plant cell. The first is centrioles, then you have plastids, then you have the cell wall, and then you have mitochondria. You know that cell wall is only present in plant cell, so this cannot be the answer. Plastids, that is chloroplast, phytoplast, etc. These are also present in plant cells and not in animal cells. So this cannot be the answer because you have to point out the organelle which is not present in the plant cell. Please read the questions very carefully. Sometimes they ask you not present, sometimes they ask you present, and people get confused. They mark the opposite answer. Now, mitochondria is also present in both plant cells and animal cells because it produces energy. So the answer is centrioles. Centrioles are not present in plant cells. Centrioles are structures which are involved in cell division. We are going to study this when we study cell division. So centrioles are not present in plant cells. Coming to the next question. Granular endoplasmic reticulum. Now what do you mean by granular endoplasmic reticulum? The endoplasmic reticulum which has granules. Or here what are they referring to? Yes, the rough endoplasmic reticulum. So granular endoplasmic reticulum is nothing but the rough endoplasmic reticulum. So they're asking granular endoplasmic reticulum is none of the above. Okay, so this option should be the last one. The other options are smooth endoplasmic reticulum which is deposited proteins, smooth endoplasmic reticulum which is deposited with nucleic acids, and smooth endoplasmic reticulum which is deposited with ribosomes. So what do you think should be the answer? What is exactly the rough endoplasmic reticulum? The rough endoplasmic reticulum is nothing but the smooth endoplasmic reticulum covered with ribosomes. Right? So the answer should be smooth endoplasmic reticulum which is deposited with ribosomes. So let's move on to the next question. Next question is, grana lamellae are sites of mutation, variation, photophosphorylation, or the dark reaction. So remember we studied grana lamellae and stromal lamellae in chloroplast. So what happens in the chloroplast? Which process occurs in chloroplast? Photophosphorylation. So the answer is photophosphorylation. The next question. Mitochondria are not found in So which of the following cells do not have a mitochondria? Mature erythrocytes, young erythrocyte, a cell, head of sperm. What do you think should be the answer? We have discussed this when we were studying mitochondria. A mature erythrocyte is nothing but a mature RBC. Young erythrocyte is a young RBC. A cell cannot be the answer because you know animal cells, plant cells have mitochondria. Head of the sperm has a lot of mitochondria. Why? Because the sperm has to swim and the energy for that swimming comes from ATP, which is produced in mitochondria. So the answer here is a mature erythrocyte. The next question. Cell organelle which is present only in animal cells. So here they are not asking not present. They are asking which is present in animal cells. Is it mitochondria or centrosome or nucleus or chloroplast? They are asking a cell organelle which is only present in animal cells. So mitochondria, as you all know, is present in both animal and plant cells. Nucleus is present in both animal and plant cells. Chloroplast is only present in plant cells. So the answer is a centrosome. Cell theory was pronounced by. So they are asking who gave the cell theory. Is it Schleiden and Schwann, Lederberg and Lederberg, Beadle and Totem, or Watson and Crick? So the answer is Schleiden and Schwann. I hope all of you know this answer. Schleiden and Schwann were the two scientists who gave the cell theory. What did Watson and Crick give? So Watson and Crick gave us the double helix structure of DNA. And their studies were based on the works, the X-ray diffraction works of a very important person, very important scientist. She was a lady. Her name was Rosalind Franklin. So by studying the X-ray diffraction studies of Rosalind Franklin, Watson and Crick were able to deduce that DNA has a double helix structure. So I'm going to tell you here, Utkalp Digital Classes is going to come up with a podcast series. The first podcast in biology deals with the life of Rosalind Franklin, the life and the works of Rosalind Franklin. Please listen to this podcast. It's really interesting. The next question. Mitochondria can be separated by. So they are asking which of the following processes is responsible for is involved in the separation or isolation of mitochondria from other cell organelles. Is it electrophoresis or centrifugation, or electrophoresis and centrifugation, or cell lysis? Cell lysis is nothing but breaking open the cell. So electrophoresis, you all know. You must have studied agarose gel electrophoresis or polyacrylamide gel electrophoresis. And centrifugation also you know. So here the process involved is centrifugation. Using a density gradient centrifugation, the different organelles of the cell can be separated. So these organelles have different densities and they get separated using the process of centrifugation. The next question is, mitochondria or chondrosomes were discovered by. So who discovered mitochondria? So the answer here is Benda. Carl Benda was the one. He actually did not discover mitochondria, but he named mitochondria. So the answer is Benda. Moving on to the next set of questions. Which of the following statements about mitochondria is not correct? They serve as sites for cellular respiration. They are enclosed by a double membrane. They are the sites where most of the cell's ATP is produced. And they are found in animal cells only. Plant cells have chloroplasts instead. So what do you think is the answer? This is very easy. Mitochondria is the site of cellular respiration. So this is correct. They are enclosed by a double membrane. This is also correct. They are sites where most of the cell's ATP is produced. Yes, this is also correct, and that is why they are called the powerhouses of the cell. So the statement which is not correct, and the answer is they are found in animal cells only. Plant cells have chloroplast instead. This is wrong. Plant cells also have mitochondria. All of the following processes occur in endoplasmic reticulum except. So which one of the following does not occur in endoplasmic reticulum? Urea cycle, fatty acid elongation, fatty acid glycosylation, or secretory protein synthesis. Protein synthesis occurs in the endoplasmic reticulum. Fatty acid elongation and fatty acid glycosylation also occur in the endoplasmic reticulum. So the answer here is urea cycle. Next, new proteins destined for secretion are synthesized in the Golgi, free polysomes, nucleus, or the rough endoplasmic reticulum. What do you think is the answer? Yes, both Golgi and endoplasmic reticulum are somehow involved in protein synthesis and secretion, but the synthesis of secretory proteins occurs in the endoplasmic reticulum and their modifications occur in Golgi. So the answer to this question is the rough endoplasmic reticulum. Nucleus is not involved in the translation of proteins or the synthesis of proteins. It is involved in transcription and replication. The formation of the replication of DNA and the formation of RNA. Now, the next question. All the following enzymes are found in lysosomes except. We studied this when we studied lysosomes. So, which of the following enzymes are found in lysosomes? Can you tell me? Acid phosphatase, yes, it is present in lysosomes. Lipoprotein lipase, is it present in lysosome? No, it is not present in lysosome. So the answer is lipoprotein lipase. Cathepsin is another enzyme which is present in lysosomes, and N-acetyl hexosaminidase A is a very important enzyme of the lysosome. Can you tell me why? We have studied this. So in the absence of N-acetyl hexosaminidase, people suffer from the Tay-Sachs disease. What is this Tay-Sachs disease? So this enzyme, the N-acetyl hexosaminidase, it is responsible for the cleavage of cerebrosides and gangliosides. If this enzyme is absent, there is an excess accumulation of cerebrosides and gangliosides in the neurons, due to which mental retardation can occur or death can also occur. So the absence of N-acetyl hexosaminidase is referred to as the Tay-Sachs disease. Coming to the next question. Rough endoplasmic reticulum is associated with. We have discussed this just now. What is it associated with? Lipid synthesis, protein synthesis, nucleic acid synthesis, or vitamin synthesis? Yes, it is protein synthesis. The maximum size of a cell is limited by. So which factor limits the maximum size of a cell? Its need for enough surface area for exchange with its environment, the number of organelles that can be packed inside, the materials needed to build it, or the amount of flexibility it needs to be able to move. What do you think is the answer? So the answer is option A, its need for enough surface area for exchange with its environment. Next question. You would expect a cell with an extensive Golgi apparatus to do what? So there are 4 options. To make a lot of ATP, secrete a lot of materials, move actively, or perform photosynthesis. So if a cell has extensive Golgi apparatus or a lot of Golgi apparatus, what will the cell do? Golgi apparatus is involved in secretion of proteins or secretion of materials. So the cell is expected to secrete a lot of materials. I hope this is clear. So if a cell is expected to make a lot of ATP, which organelle is going to be in excess? It's the mitochondria. Move actively, it is going to be the cytoskeleton. And what about photosynthesis? The cell will have excess of chloroplast. Moving to the next question. Which of the following correctly matches an organelle with its function? So organelles are given to the left hand side. You have to match them. You don't have to match them. There is one organelle which is given correctly with its function. All the others are incorrectly matched with their function. So you have to identify which organelle is correctly matched with its function. Mitochondria, photosynthesis. Is this correct? No, it's not correct. Nucleus, cellular respiration. Is nucleus involved in respiration? No. Which organelle is involved in respiration? It is the mitochondria. Which organelle is involved in photosynthesis? It is the chloroplast. Ribosome, manufacture of lipids. Does ribosome manufacture lipids? No. Ribosome manufactures proteins. So the correct option is central vacuole, which is involved in storage of materials. Moving on to the next question. Golgi apparatus of plants and lower invertebrates are usually referred to as. So what is the second name of Golgi apparatus in plants and lower invertebrates? Is it desmosomes? No, it's not desmosomes. Is it dictyosomes? Yes, it is dictyosomes. So the Golgi in certain plants and lower invertebrates is also known as dictyosomes. Next question. In eukaryotic cell, the region between the nucleus and the plasma membrane is called the... So this is a eukaryotic cell. This is the plasma membrane. This is the nucleus. So the region between the plasma membrane and the nucleus, what is it called? The cytoplasm. What is nucleoplasm? All the fluid inside the nucleus is called the nucleoplasm. Next question. In muscle cells, the dash is specialized for the storage and release of calcium. Which organelle is specialized for the storage and release of calcium? We have studied this. It's the smooth endoplasmic reticulum. So smooth endoplasmic reticulum is involved in the regulation of calcium levels in the cell. When the cell has depleted calcium levels, certain channels open in the membrane of the smooth endoplasmic reticulum and the calcium comes from the lumen of the smooth endoplasmic reticulum into the cell. And when there is an excess of calcium into the cell, these channels again open reversibly and the calcium from the cell enters the smooth endoplasmic reticulum. The next question. Of the following organelles, which group is involved in manufacturing substances needed by the cell? Which group is involved in manufacturing of substances? So the first is lysosome, vacuole and ribosome. This is a tricky question. Even if you get one organelle wrong, you will get the entire question wrong. So think about the functions of each organelle mentioned in the group. What does lysosome do? Lysosome is responsible for autophagy, digestion of certain substances in the cell, right? So lysosome is not responsible for manufacturing substances. So this option is incorrect. What do you think? Is ribosomes involved in the manufacture of something? Yes, proteins are synthesized on the ribosomes. The next is rough endoplasmic reticulum. Yes, RER also synthesizes proteins. It is the ribosomes which sit on the RER that can synthesize proteins. Obviously, free ribosomes in the cytoplasm also synthesize proteins, so ribosome is also correct, and rough endoplasmic reticulum is also correct. Do you think smooth endoplasmic reticulum is responsible for the manufacture of substances? Yes, smooth endoplasmic reticulum is also involved in the manufacture of certain types of proteins and glycoproteins. Vacuole again is incorrect because vacuole is a storage organelle. Rough endoplasmic reticulum and smooth endoplasmic reticulum are correct, but since vacuole is incorrect, this answer is not correct. This answer is also not correct. So the correct answer is B. Now, which of the following molecules moves regularly from the nucleus to the cytoplasm? What moves from the nucleus to the cytoplasm? This is a very easy question. Is it glycogen? Is it RNA? Is it DNA or cholesterol? The answer is RNA. So nucleus has DNA which gets replicated and forms multiple copies of the DNA. This DNA then forms RNA by a process called as transcription, and this RNA leaves the nucleus and enters the cytoplasm. Here in the cytoplasm, it is translated into proteins. So it is the RNA that moves outside the nucleus into the cytoplasm. Next question. Lysosomes contain enzymes capable of what? Is it aerobic cellular respiration? Is it digesting a part of the cell? Synthesizing proteins or synthesizing lipids? What do you think it is? Yes, it's digesting a part of the cell. Lysosomes are digestive organelles. Which subcellular fraction of mammalian hepatocytes? Hepatocytes are nothing but liver cells. Which subcellular fraction of mammalian hepatocytes contains the enzymes necessary for elongation of long-chain fatty acids? Though the question seems a little tricky, but it's very easy. A subcellular fraction means when you subject a cell to density gradient centrifugation or fractionation, which subcellular organelle or which organelle simply is responsible for the elongation of long chain fatty acids. So in simple words, they are asking which organelle is responsible for the elongation of long chain fatty acids. We have just discussed this. This is endoplasmic reticulum. This was all. I tried to discuss all the important questions of Unit 2. But mind you, this is not enough. You have to go back. You have to practice a lot of model test papers or previous year papers. Find out questions from cell biology and practice them. Subscribe to Utkalp digital classes for all the latest content on CSIR NET Life Sciences. Please do not forget to press the bell icon. This will keep you up to date with the latest notifications. And please go back, do a lot of previous year papers, revise a lot of units, time your preparation. This is what I've been telling you again and again. Timing your preparation will lead to the qualification of the exam. Thank you.